Very good morning, everyone. So here, on behalf of Kerala Startup Mission, GTech group of companies in Technopark, Trivandrum, Thai Kerala, and Kerala Technological University, I would like to welcome Dr. Gautam. Dr. Gautam is clinical director at MyLab's Discovery Solutions. MyLab was the first company in India to get recognition and approval for commercial manufacturing of PCR-based COVID test detection kits. It's been a really proud moment, sir, to have you here today for this session, and we are quite eager to listen to you, listen to the success story of my lab, and it's been a very phenomenal journey, I believe, for my lab, and it gives every Indians a sense of pride that we have one company from India who has been a pioneer in manufacturing PCR based COVID test detection kits in India. And being a person from Kerala, we are double proud because what I read from newspaper is that our MP Dr. Shashi Tharoor sir had in fact procured MyLab kits to Trivandrum constituency. I think that was the first
Yep.
uh, procurement by a member of parliament, and I suppose that is the first anywhere in India, any state which has procured. That was the first time for the test, if I'm not mistaken. So,
Right.
once again, sir, on behalf of Kerala Technological University, Kerala Startup Mission, group of technology companies, GTech and Thai Kerala, and all faculty members, students who are listening to this session. Thank you so much for being kind and graceful for agreeing for this session. Over to you, sir.
Okay. Thanks a lot, Arun, first of all, and good morning to everyone who have taken time out for this session. So, obviously, I'd like to thank first of all the Kerala Startup Mission, TAI, the GTech companies, and the Kerala Technological University. So, as Arun just pointed out, that as a company, MyLab has come into focus over the last 1.5, 2 months for this COVID-19 testing. But the idea of today's session is that it's not only what we have done over the last 1.5 months. Obviously, all of us know about MyLab and the COVID-19 testing because of the pandemic. But as students or people who, of some of you might be looking at the startup infrastructure, you might be part of startup, is that what we need to understand is this is a long journey. We started a long time back, and this is just one part of a continuing journey. We obviously never hoped or never knew that something like this would hit our country or hit the world in 2020, but it's there and we are facing it right now. So as people from scientific background, or even if you're from any industrial, technical, or engineering background, it's important to have your background, your base prepared, so that if there's any crisis like this, we can straight away make use of the base and then help our community, our country, and probably why not? We could be the world leaders. We could help the world also. So this is what we'd like to understand today: is that how MyLab was able to do what it did in the last 1.5, 2 months, the journey, the story of MyLab, and a bit of glimpse into the future as to what MyLab is planning to do over the next 2, 4, 5, or 7 years. Right. So we can move to the next slide, Arun. So, who are we? Basically, MyLab Discovery Solutions. It's a company which is based in Pune, registered in Pune in Maharashtra. We have a manufacturing unit in Lonavala. So, we are mainly a biotech company, and we specialize in developing and commercializing molecular diagnostic kits. Molecular diagnostic kits means, obviously, as most of you would understand, kits for diagnosing an infection or a disease or any condition based on the molecular structure or the molecular sequence of that particular organism, or it could be anything. So it's not something wherein we only restrict ourselves to clinical diagnostics. Obviously, clinical diagnostics is the most commonly heard uh, you know, thing that we are involved in, especially coming after the COVID thing. But it could be drug discovery, looking at biomedical research, so whether a particular drug is working on a particular organism or not. So with the help of molecular diagnostics, you can grow the organism and the drug, and after a few days, you can look whether that organism has increased in numbers or not. And if you're using molecular techniques for that, that's the most sensitive way of trying to understand whether a drug is working or not. The same thing applies to cancer also. A lot of newer drugs act on a particular mutation of a cancer. So if a drug is working, then the number of cells with that particular mutation should go down. So that's where drug discovery and biomedical research make a lot of use of molecular diagnostics. One very important factor is agrigenomics now, with a lot of these genetically modified GM crops now being spoken about, and some problems also arriving out of it. It's very important that countries or companies who are very quality conscious test for the presence of certain GM crops, because some of them have been known to cause diseases in human beings. Some of them have been known to be of less nutritional value, and we don't want our food to be having that particular GM crop, or ingredients from that particular kind of source. So it's important that molecular diagnostics is used to identify the presence of those GM crops, and finally see if we can have a very good QC based on that. The same thing applies to animal and food safety. It's the mixing of meats. We want to ensure that a particular species of meat is not being consumed. This could be for religious reasons, this could be for regulatory reasons. And that's why we have kits to identify particular meat species, so that we ensure that whatever food is imported out of India or exported out of India, or certain countries, if they are importing a particular kind of food, it's not adulterated. So it's a huge group of products that we cover. The main focus today of this talk, and even for the company, has been clinical diagnostics, but we consider others to be equally important, and we should be able to contribute. Next slide, Arun. So let's concentrate on the journey. As I was telling you, we we were able to contribute to this crisis, and a lot of other companies in India are doing it. So we are all proud for all of us. There's no two thoughts about it. But what's important is that the we have to understand the journey behind it. Okay, the destination today for us has been a PCR based COVID test. That's great, but the PCR based COVID test did not just come into being. We as a company have been working hard. A lot of other companies have also been working hard. It's been a long journey, and the idea of today's talk, especially for students and for the startup people who are running startup enterprises over here, would be to try to understand this journey, see if we can learn anything from this, and probably 4 or 5 years down the line, we might have, maybe lesser than that, we might have 30 or 40 companies like MyLab contributing to whatever crisis India is facing at that point of time. So this is the idea of today's talk. Let's go ahead. So the company MyLab Discovery was founded by Hasmukh and Shailendra. So Hasmukh and Shailendra both have a background of biotechnology. They were working with multinational companies. As I think a lot of us have get a first-hand experience of how to run a successful enterprise by working with multinational companies, because probably they have had the kind of startup infrastructure and environment that we are seeing in India for the last 5 to 7 years. They've had that 30 or 40 years back. So from that experience, we learn that how to set up a company, how to ensure that the standards are maintained, regulatory, quality, all these are maintained, and then we can start our own enterprise. So how did it all begin? So both a lot of us were from clinical diagnostic background, but majorly people who were looking at molecular science in India until 7 or 8 years back would know that there was very limited use of molecular biology in clinical diagnostics. Molecular biology was mainly restricted to research labs which were doing sequencing, labs which were trying to understand the genetic causes of various diseases, but they were not trying to use, or until that time, 7 or 8 years back, the application of molecular biology in clinical diagnostics was limited, and especially in our country. Molecular biology has been in use for managing diseases and controlling the spread of epidemics in the West for about 15, 20 years. So it's a comparatively new field. Yet India was still lacking behind. There were very few labs or very few hospitals in India which had adopted these techniques. So there were a couple of reasons for that. Number one, the expertise. people who were doing molecular biology were mainly restricting themselves to research and working in academic institutes. The setup cost of a molecular lab was very high. Even today, the cost of a real-time PCR is about 15 to 18 lakhs, and that's a very basic instrument. If you talk about a slightly higher advanced instrument like NGS, next generation sequencing, that goes into maybe 50 lakhs or maybe a crore. So the the ability to set up molecular labs was very few. So what happened in that case was whichever hospital started using molecular techniques, were the really high end, five-star hospitals, as we might call them. I'm not taking names over here, or they were the very well-funded central government or some few top institutes of the country which had a molecular lab. It kind of became a high-end luxury product. The idea was to ensure that we have to take these solutions. The application of molecular biology has to be taken to the common people, to virtually everyone, especially when it comes to cancer and infections. These are the main focus areas. There are so many pathogens who have been completely whose genome has been completely found out now. So we can identify any particular pathogen, bacteria, virus, fungi, based on a particular gene sequence. Even with cancers now, we know which particular mutation is causing which particular cancer. So I think these two are the major applications for molecular biology. And I think the world was changing, and it was time to India for change too. That's what something we realized 4 or 5 years back, and we started our process. Next slide. So the first thing we did was we established an R&D lab, a research and development lab in Pune itself in 2015. We got it approved by DSIR. The next thing we started doing was we started developing and validating a few real-time PCR assays. Real-time PCR assays are important because, as I was telling you, an RT-PCR instrument is still found in a lot of labs these days. So if you develop an assay which is based on real-time PCR, larger number of labs can adopt it. So that's what we started doing. We obviously started providing these assays to a few of the academic bodies like National Institute of Reproductive Health, NARI. We also went to National Institute of Cancer Prevention, a few of these institutes which were looking at newer assays to ensure that they were able to detect newer diseases, prevent spread of certain diseases. So the idea at that point of time was, we were still restricting ourselves to the academic bodies. And at that point of time, we realized that, okay, one part of the entire process is, going to academic bodies, research institutes. But at the end of the day, we have to ensure that we have enough production, we have enough capacity to start providing these kits to regular labs, to the SRLs, to the Lal Path labs, to the metropolises of the world. And to do that, we needed a proper, full-fledged manufacturing facility. So we understood sometime late in 2015, early 2016, that the time had come to move on from purely being an R&D lab-based product manufacturing, wherein what we used to do was we used to develop an assay, give it to a lab, and call it as a RUO product, which is a research use only product. The lab would then validate that test, run a few assays, and then start doing laboratory-based assays based on the assay we had provided them. So that was the basic way of doing it, and that's how most of the labs in molecular diagnostics work. But our plan was to move into full-fledged manufacturing. And let's go to the next slide. And that's how sometime around 2016 we zeroed down on a manufacturing facility. It was not a molecular diagnostic or a healthcare manufacturing facility. We just went to Lonavala industrial area, which is just about 50 or 60 kilometers from Pune. It's a beautiful hill station. And then we zeroed down on a unit which was virtually defunct, not working at all, and we started working on it. So obviously, in order to get a healthcare manufacturing unit going, we had to undergo a lot of learning. We also learned a lot, and we also had to go to a lot of authorities, right from getting approval from the pollution control board to ensuring that there was a continuous supply of electricity. We had to go to FDA for all the possible licenses, and that's how we started working on establishing a full-fledged manufacturing facility. Once we had this facility, we started sending samples to CDSCO, which is Central Drug Standards Control Organization, for designated laboratories. So the way the system works is, the first thing that we do is, for all manufacturers, or probably all students over here, the way manufacturing licenses in India work for diagnostics is that first you develop an assay. Once you are confident that you can develop it and you can send it to clinical labs, you approach the CDSCO, or the Drugs Controller General of India. Then they give you something what is known as a test license. A test license is nothing but a license which allows you to manufacture certain batches of kit which have to be given to designated laboratories of CDSCO. They're not That is not a commercial manufacturing license. That is purely a license to manufacture test batches. So once we get these test batches, based on the standards given out by CDSCO, some of them want to send it, and they also have designated labs. So they would tell us, say, send 500 samples of 3 batches of particular test to a lab in Noida, which is a National Institute of Biologicals. So that's how it works. In case of COVID, say it was Nari or it was NIV Pune. So CDSU tells us which particular lab the sample has to be sent. Based on that, they give us a test license. We manufacture those batches, get the quality and everything checked, and we send out the batches. What we were doing simultaneously was we were also carrying out multiple field trials. The reason being, it's one thing to send a few tests to one particular lab, but it's important whenever you develop a test, it's important to do multiple field trials. Probably sending one batch in a very, very temperature controlled manner to one lab in Delhi from Pune is easy. But if someone tells me that you have to send a batch to somewhere, say in Assam or Shillong, because then the logistics are different. You have to ensure that the temperature is maintained as the tests are carried from Pune to that particular center. Then we also have to ensure that once they reach that particular center, are the tests being carried out properly? So while the regulatory requirement was just to send samples to one lab in Noida, we were also sending it to other labs across India. In Bangalore, we were sending it to Nagpur and Delhi, other places, and we were asking our labs who are friendly with us not to use these tests for any diagnostic purpose, but as and when you get the time, run these tests in parallel to the gold standards that you have, so that we get to know, if we get a first hand feedback from you, is our test meeting the standard of those imported kits that these labs were using. So we were getting feedback, and luckily for us, because of the work done by the R&D team, whatever feedback we were getting prior to that, prior to the CDSO approval, was good. People were saying your kits are able to match the standards of whatever imported kit for whatever diagnostic test we are using. So we were doing a lot of groundwork that point of time. Yeah, Arun. So based on that, as I was telling you, we submitted our kits to the CDSCO. Uh, at the same time, our facility was inspected by FDA, which is the Food and Drugs Administration, because manufacturing comes under the local state government. The sites were audited, we got the ISO license, we got all the possible clearances. And finally, at the end of 2018, we got our first manufacturing license. And the first license that we got was for HIV, HBV, and HCV quantitative assays. So people who are infected with, say, hepatitis or HIV, they undergo treatment for these diseases. What's important is clinicians want to understand if a particular patient is under the treatment, say, to control HIV, is is the count of HIV in that person's body increasing or decreasing over a period of time? So that's an HIV quantitative assay. This is considered a very, very critical diagnostic test. So, of the manufacturing licenses, Class C and Class D are the most difficult ones because they are critical assays. And we got a license for Class C and Class D. Similarly, very soon we got a license for NAT. Now, NAT is Nucleic Acid Amplification Test for blood donors. We'll come to that a little while later. So that's how in 2018 or 19 we got the licenses for these tests. Uh, we obviously are carrying out work for other tests, and today we have received test license for more than 100 products. So obviously, over the last 3.5 months, we have been stuck, or we've been working very hard on the COVID. But in the background, we're working on more than 100 products, which will be launched over the next 6 to 8 months, or the moment this crisis is over. So this is just a slide of those essays that we managed to get. So on the left is the HIV, HBV, and HCV quantitative assays. On the right is the ID-NAT. So ID-NAT, as I was telling you, is individual donor nucleic acid amplification test for HIV, HBV, and HCV. So let's go to the next slide, Arun. Yeah. So the ID-NAT is used for screening of blood donors for these infections. Our ID-NAT, the MyLab ID-NAT, the name of that is NatSpert, is Asia's first and world's second ID-NAT approved for regulatory bodies. So why is ID-NAT such an important test? What happens is, whenever you are suspecting a patient to have a disease, you use something known as a diagnostic test. You know that this person has a disease or most probably has a disease, and then you test for the presence of an infection. Say, let's to simplify the matters, let's look at COVID. The testing of COVID is done on patients who either have some kind of symptom or who have been exposed to a patient who might have had COVID. So we have uh, our target audience in this case is suspected of having a disease. In case of ID-NAT, it's a screening test. So it's done for people who go to blood banks and donate blood. Now the idea is that all these people who go by default are healthy individuals. We are not suspecting them of having any of these HIV, HCV, or hepatitis B. Even on taking history, we ensure the blood banks ensure that these patients are not having a bad history. So when we are using a test to screen for people for the presence of such disease, the tests have to be very, very sensitive. So the viral loads at which you want to detect the presence of these infections is very low. So this is something which that we are very proud of. We are this, as I was telling you, we are the second company in the world. It's one of the most sensitive tests that we have developed, and more than 1 lakh people have already carried out the testing across the country now at multiple hospitals. We've had a lot of publications coming out. We can go to the next slide. And apart from these particular tests, there are about 140, 150 labs across India which are using our other assays. Some of them are CDSU approved, some of them are under RUO. So a lot of tie-ups we've carried out with leading hospitals, healthcare institutes, or academic bodies. And we're also looking at developing new tests. New diseases are being told to us that, why don't you work on a particular new syndrome that has come. So we keep on adding new tests to our kitty. We keep on getting new test licenses. Yeah, let's go to the next slide. And obviously, as time went on, we also expanded our team. So obviously, Hasmukh and Shailendra were the first ones in 2014-15. Now we have about 5 or 6 directors and a big team now. And the team is a combination of R&D, clinical diagnostics, clinical medicine, regulatory, finance, application support. So it's a huge mix. And this is something that, as students or people from the startup background should also understand is that a team cannot be formed by just one specialty. Even if you have the greatest of brains to develop a new product, we would definitely require a marketing or a sales team to ensure that if you have developed a product, that product is sold to the right people. It's very important to have an application support team because even if that product is great, it goes, the best of products require some kind of support to be given out. So it's important then to have an application support team. It's important to have a person with a nice financial background because see at the end of the day selling and manufacturing and support is one part, but one person should have proper control over the finances. What are the budgets required? How do we plan for the next quarter? How do we plan for the next year? So I don't think any company, anything in the world can just work with one or two expertises to run any successful commercial enterprise or any enterprise for that matter. It's a group of people, and we are lucky that now we are a group of 5 or 6 directors and a much bigger company now. And I think all of us are contributing in our own special little way to ensure the company does well. Yeah. So as I was telling you, we've also concentrated a lot on the academics. So it's not only purely a commercial company. The idea is to obviously generate commerce, but uh, there's something known as evidence-based medicine. So whatever you are doing, it's important for that to be published. It's important for the peer groups to understand the application of that particular whatever test you've developed. So we have now published a lot of cases in 3 or 4 international journals. I've just put a couple of them in front of you. This particular one on the left was something we published in the Global Journal of Transfusion Medicine. On the right is a presentation or something which was published for ISBT Barcelona, which got postponed because of COVID. So it was supposed to be presented in June, but now it's been rescheduled for December. So the idea again here for all the people listening is that one part of it is making and selling a particular drug or any particular product. But it's important to then use the background that you have to publish what you've done, because that's the only way you can get endorsed. That's the only way you can get recognized, and that's what a lot of companies, if you look at the big companies in the world, the Abbotts, the Roches, they ensure that whatever scientific work they are doing, yes, commercially important, but it's also being published, it's also being compared, it's also being read by the peers. So, and that's how the good news about the product spreads. Yeah, next one. So apart from the products, we started developing our own automated systems also. So apart from the purely the kits, we realized that it's important to provide India or the developing world with an automated system, because one part is understanding the test, the other part is the ability to carry out the test. So there is a shortage of trained manpower. So it's important to give fully automated systems a good platform for people to carry out the tests. Yeah, let's go on. So now let's come to the main issue, which is coronavirus and the way we responded to it. Yeah. So basically, a little bit about coronaviruses. It's not something which has come recently. It's been known. It's the first time it was identified was somewhere in 1960s, and it's usually there are four main families: alpha, beta, gamma, and delta. And out of these, the beta coronaviruses are known to cause human diseases. They're mainly found in animals, but quite a few of them are known to cause human diseases. So until 2019, I think six out of this huge family were known to cause human diseases, and SARS and MERS, which is the severe acute respiratory syndrome and the Middle East respiratory syndrome, I think 2003 and 2010 or 11. These were the ones which had caused a lot of hue and cry in the last 2 decades. Again, they were all coronaviruses. The when the crisis started in late 2019, early 2020, people found out that a lot of people were developing pneumonia-like symptoms. And with the help of NGS, they were able to find out that it was a type of coronavirus, but it was slightly distinct from the SARS and the MERS, which is causing it. And there were 6 or 7 strains which were available in the public domain by that time. So what happened was that various institutes across the globe, they started developing their own assays to identify these various strains which were causing this particular type. So there was CDC in Atlanta, there was the Charité in Berlin, which was a WHO approved facility, which also came out with a particular protocol to detect the particular infection. Let's go ahead. So let's just try to understand. I mean, just for I mean, even though this is recent history, but it's very important to understand how what happened. So I think it was on 31st December 2019 that China actually told the world that there is a group of people in a particular province which are showing a lot of pneumonia-like symptoms. And WHO obviously responded very fast. They set up something known as the Incident Management and Support Team to look into this particular outbreak. By 10th of January, WHO realized that yes, this looks like a bigger problem, and they came out with a comprehensive handbook on how to detect and how to test. By 12th of January, China made the sequence of that particular virus, the genome or that particular type of coronavirus public, okay, which was causing this particular pneumonia. And many centers, as we just discussed in the previous slide, many centers across the world recognized this new strain and they started developing uh, tests for coronavirus. So this was all happening just about, less than 4 months back. Next slide. Well, as far as MyLab was concerned, so we, like all of us in the world, we really didn't know how big could this be. In fact, we as a company were very busy with a big blood transfusion conference in Mumbai in early January, from 9th to 11th of January. At that conference, we actually uh, you know unveiled or showcased our upcoming automated system to the world, and we were really looking forward to launch that system in March, with final validations to be carried out. At that point of time, we, around 15th or 18th of January, or a little later than that, we realized that, okay, it seems like there's something big which is happening in China, or at least in Europe at that point of time. And maybe it's important that, we should also prepare something. Why don't we make use of our expertise? Why don't we make use of our R&D lab, of our manufacturing facility, of the sources of the primers, probes, and every information that we have? And why don't we also start developing a test for COVID-19? What if it turns out into a bigger problem? So we started working around it around sometime in the middle of late January. Next one. So basically, what we did, as I was telling you, there were a couple of labs. There was a lab in CDC Atlanta, which is run by the US government. There was a WHO lab in Berlin, which was carrying out protocols, which was giving protocols for testing. There were protocols coming out from China. So obviously, we had to zero down, a bit of guesswork, but a lot of academic work went on for those 10, 15 days. We decided, okay, let's follow the WHO protocol from Berlin to use, that particular uh, screening method, which is the E gene and the RDRP gene. So that's how we zeroed down on a particular sequence. We designed the primers and probes. We had a lot of discussion over whether to go for singleplex, whether to go for multiplex. One very important factor that all of us have to understand was the lack of the availability of a known positive sample. If you look at it in late January or early February, there were just a few cases in India. So as a manufacturer, even today, it's impossible to get hold of a known positive sample. So, as a manufacturer, we had to then find out a source of the synthetic, COVID-19 or the SARS-CoV-2 RNA controls, which have the same sequence as the real virus, but they don't carry the infectivity, or they're not true viruses, but they just have the same sequence. So based on that, we were able to develop a first batch of essays. We had to get the right balance between the sensitivity and the specificity. We had to ensure that the limit of detection was right. There was reproducibility in the test. There was no cross reactivity with other similar infections like H1N1 and HIV and others. We also had to ensure that there was stability on these assays. So once we were able to do that, we were confident within ourselves. We knew that this particular lab, this particular test has reached a particular standard. Let's go to the next slide. Yeah. Meanwhile, what was happening with coronavirus in India and across the world? So the first case was reported, I think, on 30th January from the state of Kerala, and I think there were just two more cases reported in February, and all of them were students who were returning from the Wuhan province of China. So luckily for India, there was no major outbreak in January or February, which was a great thing because we got the right time to prepare. Obviously, the numbers were increasing significantly in the rest of the world. In fact, by early March, I think WHO declared COVID-19 to be a pandemic. More than 20,000 people and more than were infected in Europe. 1,000 people died in Europe by end of February. So in India, we were a little lucky in that sense because the virus took a little time to come and spread. Because probably it was the proactive steps of the government. We were ahead of time in that case. So the confirmed cases of India reached just about 100 by 15th of March. So what happened was by that time is that we got the right amount of time to start and developing and validating the assay. Let's go to the next slide. Yeah. And by first week of March, we were ready with our essay. We were confident. We knew that we've come out with a great product. We approached DDC, the Drugs Controller General of India, and we got a test license. Now, here is something I would like to spend a minute or two over here. The government of India did a wonderful job. They normally, when you approach government bodies and you ask for a test license, they would take week, 10 days if you're lucky to give you a test license. Things were happening in hours over here. They were not bypassing they were not giving any shortcuts. They were just ensuring that if as a company, as a manufacturer, I went to drugs control and told them that I have an essay ready. So within an hour of that or within a day of that, they gave me a test license. That which means that okay, if you're confident, here's the test license, take your batch and get it tested. So on 8th or 9th of March, we gave off our batches to ICMR for validation. As I was telling you, we were conscious, we were confident, we were anxious, yet we were confident of getting a good evaluation report. So until that time, what was happening was the Indian government was was completely dependent on the tests which were supplied from WHO from the foreign manufacturers. So what they were getting was a combination of primers, probes. They had to procure controls separately. They had to procure the positive and negative controls separately, and enzymes separately. So it was like a DIY kit, do it yourself. So a lab which was doing it had to make its own assay from the various components that they were getting. And obviously, while all this was going on, the number of tests in India was increasing, on a daily basis. Unlike January or February, by March we were getting 10 or 15 cases every day. So this was what happening in the background, and we were waiting. Next slide. Finally, on 23rd of March, the ICMR came out with the results. So apart from us, there were about 8 or 10 other manufacturers which had given the kits for evaluation, and obviously the evaluation process went on for 7 to 8 days, which is normal. And finally on 23rd of March, ICMR came out with the first results, and there we are. We became the first company in India to have got the proper approval from ICMR, along with one more German company. So again, we straight away, this report was sent by the ICMR to the Drugs Control Journal of India, and again, hats off to the government. Within hours of getting the satisfactory report from ICMR, on that particular day itself, the Indian government issued a manufacturing license. Again, from our experience, I can tell you that normally when a report is sent from a government authorized lab to CDSCO, that the tests are good. CDSCO would take 5 to 7 or 10 days to issue a license. In this case, they just took hours. The moment they got to know that our kits have been validated by ICMR, their ICMR has said that they're 100% specific and sensitive. Within hours of that, ICMR gave us a license to manufacture. Yeah, let's go ahead. So basically, that's how we came. So this particular kit was a ready-to-use kit. It had the combination of primers, probes, controls, master mix, enzymes, and everything was standardized. One important thing that our kit at that point of time, differentiated ourselves from what was being done, was that the screening using the E gene and the confirmation using the RP gene was carried out simultaneously. Prior to that, all the tests that were carried out in India had a 6 to 7 hour running time, wherein the first step was screening, followed by confirmation. In our kit, both the things could be done simultaneously. So within 2.5 hours of time, you were able to get the results. Our kits could be run on any real-time PCR platform. There are about now there are about 3,000 to 4,000 real-time PCR instruments across the country, and these kits can be run on any one of them. Now, very important thing that our kit had was we also put a human RNA-seq control. Let's go to the next slide to understand that. So, what is a human RNA-seq control? See, this is particular something known as the housekeeping gene. So this is a gene which is present in all human samples, irrespective of whether the person is infected or not. It's a regular housekeeping gene. It's present in all our cells. So we've also put this as a control. This helps in understanding whether the sampling has been done properly. Now, why is that important? See, unlike collecting a blood sample, collecting a sample for COVID-19 requires nasopharyngeal swabbing, which is not an easy process, number one. Then there's also the stigma associated with nasopharyngeal swabbing. So a lot of times people might not actually give you a sample. They might just give you the swab, which is not being taken from the patient's nose, and they might just send it to a lab. By having a human RNA-seq endogenous control, our kits ensure that a sample has human uh, gene in it, human print in it. Let's go to the next one. Now coming to the important part, which is okay, manufacturing it is one part. The other important part is ensuring that they reach every corner of India. So we got amazing amount of help from authorities like India Post. They sent me a personal message saying that we want to help you. Let us know where you want to send the kits. The Ministry of External Affairs helped us by ensuring that raw materials were being got into India within a matter of time. We send our kits across states, and as Arun was pointing out, the political class across party lines, Mr. Shashi Tharoor reached out to us. His office reached out to us, and he ensured that these kits were delivered to Kerala within days of us getting the license. So amazing! Hats off to all the people who were involved in ensuring our kits reach every possible state and corner. Next one. So obviously on the top left is with the help of Mr. Tharoor, we were able to ensure that our kits reached Kerala. Same thing was done by the CMO of Odisha, Mr. Naveen Patnaik. And obviously, I'm not trying to just zero down on a couple of ministers or politicians. I think everyone, irrespective of the political party they were belonging to, the bureaucrats, there were a lot of NGOs which came forward. There were many commercial, hardcore commercial organizations who dedicated their CSR team to buy kits from us and then give it free of cost or just donate it to various hospitals. So a lot of support we got. Next slide. But at the same time, at that point of time, we had a capacity. We also knew that just as we envisaged sometime in January that this pandemic may come to India, very soon, the moment we got the license, we also realized that we might need a much larger number of tests than what our capacity is. So we had to find out the right partners who will be able to help us in scaling up. And one major partner that who came with us was Mr. Adar Poonawala. He is the CEO, the owner of the Serum Institute of India, the world's largest vaccine manufacturer. They're also based in Pune. We also had Mr. Abhijit Pawar, chairman of the APG group, coming and joining us. So idea was that with the help of the funds we got from them, as Startup Mission would understand, having an idea is one thing, converting that idea into a product is very important, but having that product in the right amount to ensure that it reaches all the people and it satisfies the demand is also important. So getting a strategic partner like Mr. Adar Poonawal was very important. Next one. Yeah. Later on, we also realized that it's important to be, uh, as far as possible, be self-reliant. Something which the Prime Minister just said a couple of days back, but this is something we realized about a month back. So we tied up with Syngene, with Kiran Mazumdar Shaw's company, to ensure that we get a lot of our consumables sourced from India itself. We don't have any dependency on imported components of the kits. Next slide. Obviously, on the ground, we made tactical changes. We had to find out the right channel partners, right logistic partners. On the factory, we increased automations to increase our capacity. The application support team, that's very crucial. A lot of labs which started doing the tests in the last 2 months, they were not doing any molecular diagnostic tests at all, or they were doing 5, 10, 15 tests a day, and all of a sudden they are now doing 200, 300, 400 tests a day. So it's important for them to be provided with the right amount of application support to ensure that these tests are carried out in a seamless manner. We've also developed some online training videos so that a vast customer base can ensure that they are up to it as soon as possible. Next slide. The Department of Biotechnology again has recognized. They've also funded us for our expansion. There's a Geneva-based NGO, the Foundation of Innovative Diagnostics, which is also helping us. They're procuring kits from us and then giving it to various hospitals across India. Next one. Uh, so again, it's not only the kit. We also realized that there was something very important. Was apart from the PCR kit itself, there was the RNA extraction kit, which was in shortage. This is the feedback we got. Let's go to the next slide. I'll go a little faster now. I think we're running short of time. So we also developed our own RNA extraction kit. We got it validated, cleared by ICMR. So now we offer the complete solution, RNA extraction and the PCR kit as well, to ensure there's no shortage, no dependency on foreign manufacturers. Next one. So now coming to just touching a topic of a few cases have come up recently of false positive, false negative cases, and that's got nothing to do with one particular manufacturer. That happens with all particular kits. See, the reason over here is we have to understand that the magnitude of testing which is happening over here, large number of labs have started testing clinical samples for the first time. As I was giving you an example, labs which are purely research labs just doing 5 or 10 tests a day are now doing thousands of tests a day, maybe 500 tests a day. Not all molecular diagnostics labs have the same capacity. They don't have the same automated instruments. They don't have the same capability. They don't have the same right amount of consumables. So it's natural for some labs to have wrong results. There was a case recently wherein some pilots were given false positive results. Pilots who had flown, Air India pilots who had flown to India with the consignment. See, it could be because of poor sampling. If you have a contamination during sampling, it's possible that a large number of that particular nasopharyngeal swab was dipped in 2 or 3 different viral transport media. So it happens at times. So it's important that manufacturing a kit is one part, but it's important to have a very good QC process control of the entire testing mechanism. This could be challenging in terms of contamination and everything, but apart from one aspect, which is manufacturing, it's very important to understand the other aspects of all lab testing. Yeah, let's go ahead now. So recently, just about a week back or 4 or 5 days back, as I was telling you, with the help of Adar Poonawala, with the help of Mr. Abhijit Pawar and the DBT funding, we have increased the number of automations, and now today we have the capacity to manufacture up to 2 lakh tests a day. Yeah. Next one. So, what are our future plans? Apart from COVID-19, obviously, COVID-19 was never a part of a plan even until, say, 15th of January, right? We were obviously developing a large number of assays. We have about 100 test licenses with us. So we are planning to launch kits for cancer detection, for bacterial infection detection. In fact, one very important kit that we are developing, and it's at it's a final validation stage, is a test to detect bacterial contamination of blood components, which is mainly platelets. So until now, we only talk of blood as having potential to cause HIV, hepatitis. What we don't realize is, at least in the Western world, more number of deaths are occurring today because of bacterial contamination of blood. And we are also coming out with the first test, probably in the world, to be able to detect the presence of bacteria in that blood. So this is something we are developing. We have already developed one level of an automate, which I just showed you the image of Compact X. We are now developing a bigger automation system, a complete walkaway automation system with an integrated real-time PCR. We have got the first prototype. Hopefully by June or July, we should be able to launch that one as well, time permitting, after because of the COVID crisis. So the idea is to look at various diseases, not only COVID or blood transfusion. And coming to the last slide now, what we think is that India, especially about this particular pandemic, I think India is really capable of managing this pandemic on its own. Starting from sample collection, the PPEs, the diagnostic kits, the PCR kits, the antibodies, and everything, to medicines, to ventilators, I think we can manufacture everything. As startup missions of Kerala or various states, it's important to recognize that this is a time where India can prove to the world we don't have to depend on a single import or anything. We can manufacture and ensure that we take care of this crisis ourselves, and we can also be the world leaders. We can also export these things to the other parts of the world once we ensure that we've taken care. So I think MyLab solution sees itself as an integral part of this Atmanirbharta mission, the self-reliance mission. And I think as all of us, as students, as various parts of startup programs, the TIE and GTech, I think we should. This is something that we should have in our mind. We should try to be a self-reliant country in everything possible. Why only COVID? In everything, in terms of technology, in terms of medicines for various diseases, for cancers, everything. This is the take-home message, and I think COVID-19 pandemic is that start of the process wherein as a country we can be self-reliant. Next slide. Thanks a lot, and I'll be happy to take questions.
Thank you, Dr. Gautam. Okay, so we'll go into the questions. I hope my voice is audible.
Yeah.
Okay, okay. So, very insightful session. I think packed with
Okay.
a lot of information in the shortest possible time. So, there is this question. So, I'll go into the first question. This is from Mr. Mohit Kaushik.
Right.
So, we would like to know, like, the props and primers that you use in your
Hmm.
testing process. Are they indigenously manufactured or imported ones?
Right. So obviously, when we started, we were relying on imported, but we've also tied up, as I was telling you, with an Indian manufacturer now. So now it's indigenously manufactured, and the idea is to be completely self-reliant in the next couple of months, and we are on track of that as well.
Okay. So, since you were already involved with this COVID and pre-COVID diagnostics manufacturing process, so
Mhm.
since you have a lot of experience in facing the similar situation, so what do you think? How long would this COVID last? Because you have also been tied up with Serum Institute, and there has
Right.
been couple of I would rather say unverified reports saying that Serum Institute is going to manufacture some antidote for COVID-19.
Hmm.
So we would like to hear it from your side.
Well, see, if I mean, it's very difficult for anyone to predict exactly how long will this go on, and when will it end exactly. But I think it's going to be a multi-pronged approach. It's going to be a combination of the right to therapy, the right mix of antiviral drugs. It's going to be a preventive, proper social distancing. It's going to be, hopefully, a vaccine will come out. A vaccine usually takes 6 to 8 months, and rapid testing. So I think it's going to be a multi-pronged approach. We cannot say that, okay, on XYZ date or on XY month, a vaccine will come out, and after that, the disease will stop. It's here to stay. Like swine flu, it came in 2010 or 11, and now it's a part of our life. We get influenza jabs every year, at least not very much in India, but in the Europe or Western country, it's a common thing. So I would say it's here to stay. It will not go away, it will not vanish on July or August. What's important is we'll have a combination of therapies available, vaccines available, a good diagnostic test available to ensure that it does not hamper our day-to-day life as it is doing right now.
Okay, okay, okay. Um, if I recollect it correctly, um, Dr. Fauci of
Hmm.
US had in one of his press briefing mentioned that um, the if you wish to win a war, you should know your enemy first. That should be the first
Right.
strategy. But here in this case, even being an infectious specialist, he was mentioning that he was humble enough to accept that we still don't know a major aspect of the enemy, in the sense
Right.
like why is it like youngsters don't get
Hmm.
infected with COVID compared
Right.
to everybody. But if you look at history, we have uh, we have the previous flu, wherein which I think that it was a Spanish flu, wherein which the elderly were not impacted much compared to youngsters.
Right.
So, so how do you take it? I mean, it's quite.
Right. See, one thing is very important is to understand viruses. Viruses tend to mutate a lot. So it's, for example, I was just reading an article published last evening or today early morning in India, was that there are 6 clusters of viruses that have been found in India. One particular cluster has come from Wuhan, the other one has come from Iran, there's one which has come from Middle East or South Asia. So it's important to understand that every virus is different of these clusters, they mutate, and then based on that the infectivity is different. Now coming to the age profile, again it's a very very short time. If you look at the HIV virus, the entire medical fraternity took 7 or 8 years to understand what that virus was. It took another 8 or 10 years for them to come out with the right antiretroviral drug. Starting from 81, 82. Yes, I understand at that point of time the viral sequences were not as strong as what they are today, but it took about 15 years to come out with the right therapy. Still, you cannot prevent HIV. We know the problem, but we know how to manage it, right? But over here, we're not talking in terms of years; we're talking in terms of weeks. So every week we come out with a new strain, and I think we're doing a great job in terms of coming out, with the right management protocol. There will be some hits and misses. Nothing is going to be right on the first go itself. So as Dr. Fauci pointed out, yes, it's a new enemy. We really don't know the exact way it's going to work, but it will be a combination of factors which will help. I'm a firm believer that as a human race, in a couple of months we will be able to control it. I mean, the virus will not control us. We'll rather control it, but nothing is going away.
Okay. So, Mr. Rishad Mr. Rishad would like to know the any startup would require some strategies for product marketing.
Mhm.
So one thing which was quite favorable, I'm sorry to say favorable in the sense it may not be the right word, but then
Hmm.
the the emergence of COVID
Hmm.
perhaps provided you the right opportunity to
Right.
ensure that the set of products that you have can be customized for COVID, and to ensure that the testing process, everything happened as fast as possible without diluting the standards, as well as
Right.
the testing institute of the government communicating with other branches to ensure the earliest start of commercial production. So otherwise all these procedural functional help from the government, what were
Hmm.
your marketing strategies for this particular product?
You're talking about COVID or in general about other products?
We would like to know both other products as well
Right.
as
Right. See, in terms of the other products, the classical products that we've had a license for nearly 1.5 years, so we were following a very regular, scientific approach. So we were not. The idea was to have a lot of publications, have major institutes, uh, you know, visiting them, ensure that our product is working over there. They are talking to other in the peer groups, and more of a mouth to word of mouth publicity. We are not a huge multinational which is backed by multi-billion-dollar marketing platform. We cannot go on TVs. So the idea was very clear: believe on the product, ensure that the product works properly, ensure that the doctors or the labs or hospitals which are using up their product publish papers, talk to the peer groups, ensure that others come to know that we have a good product made in India, and that's how it'll go. As far as this COVID thing came, yes, I'm lucky. Yes, we were lucky in that sense, but we are also lucky because it could be said like this: fortune favors the brave. We were the first ones to get it approved, but we were the first ones to get it approved because we had a proper R&D lab, and we started working on it way back in January. So yes, because of that, we got a boost, and people realized that we don't have to rely on foreign-made products. Even an Indian company can also help us. And probably we believe that it acted as an inspiration for a lot of others, not only in terms of diagnostic kits, probably PPEs and ventilators, who thought that why not? Why do we have to rely on foreign-made kits? So, as far as marketing is concerned, I would still say it's when you're talking about a healthcare product, it should be very hardcore scientific approach to ensure that the word spreads. Yes, there might be unfortunate events like the pandemic, wherein if you're there at the right point of time, you might get a boost because of the solution that you provided.
So next we have a question from one of the budding entrepreneurs from Trivandrum. So as you would be knowing about Kerala, the entrepreneurial activity in Kerala, we have
Hmm.
in Kerala we had the first IT park in India. Our state started the first IT park. We had the first startup policy in the entire country, and so we also have the first biomedical incubator in the country, which is run by Sri Chitra Institute of Medical Science Technology. We have Asia's one among the biggest blood bag manufacturer company based out of
TTK.
Kerala. We have
Yes.
Dentcare Dental, which is again Asia's biggest dental prosthetics manufacturing companies. So um, so we would like to know. See, we have a good amount, as I've already mentioned, we also have a biomedical park which is coming up very soon. It has already almost completed, but it is expanding massively in Kerala, in Trivandrum, and in Cochin. So we would like to know these budding entrepreneurs in any of these areas. Would you be open for any collaboration, partnership with these companies so that on a win-win mode?
So the answer to that in one word is yes, definitely yes. But obviously, it's also important for us to understand, what product it is. It should not be. We should be able to synergize. I mean, if they have a product and we obviously now have a good presence. Again, we are not as huge. We are not a, we are not a GE or we are not a Siemens right now. But obviously, we have a good reach. And if there's a product which the budding entrepreneurs think will have application, will support the healthcare, why not? The answer to that is yes. We'll be very happy to partner.
So you had mentioned in your session that procuring the pure samples for gene
Hmm.
sequencing is really tough.
Yes. Well, the answer is not tough. It's not allowed as of now in India. So for manufacturers, we don't have access to a real clinical sample so far. In fact, we are a part of an organization which is known as the Association of Diagnostic Manufacturers of India. So we have a regular interaction. We have WhatsApp group. So we are approaching the health ministry and asking them that in the West or even in South Asian region, Korea and all, the manufacturers get access to such samples so as to develop newer kits. Unfortunately, that is not so far allowed in India, and we've got a positive response from the health ministry, and they're saying that they are looking at ways and means of ensuring that it reaches manufacturers without any harm.
So would yourself and MyLabs be looking for an academic partnership with engineering colleges or biomedical institutions in the state of Kerala?
Definitely. I mean, as I was saying, in general, we are very, very open to various ideas. If there's any particular medical college or biomedical engineering college which has an idea, we'll be very happy to share. And forget about getting an idea from them. Even if we as a company want to carry out a few modules, go to the hospital, go to the to those colleges and impart knowledge about molecular diagnostics, be it online, be it via lecture by someone coming down, we'll be very happy to do that because we believe the next generation of Indians should contribute much more than probably what my generation or the previous generation has done. With every new generation coming in, every new batch of students coming in, I think we should be contributing more than what we've done right now.
Okay. So as our lockdown is entering the next phase very soon,
Hmm.
probably by Monday, so government is looking forward to kickstart the economy at least in a medium manner.
Hmm.
But the one sector where clarity is missing, or even perhaps we don't have a precedence, would be the education side, especially
Right.
the primary classes. How
Right. Yeah.
primary school, how to go about it? Because if it is a secondary or higher secondary, you can teach the kids, you can make them
Right.
aware. But then, so the alternative that governments and regulatory authorities have is let's have online classes. But then
Yeah.
it is not really feasible for small kids, let's say LKG,
Yeah. I
UKG.
understand.
So, what is the way forward?
See, purely in terms of the concept of a classroom, that is a classical case of the chance of spread of corona. It's a small room where you have 40 or 50 children sitting together. So, fortunately or unfortunately, that is one area that has to be addressed, and that's where probably not nothing from my side, but the GTech, which is one of the sponsors of this particular thing, technology has to come in. Yes, children of a particular age may not be able to understand. It's okay for a 15-year-old to get an online class, but how do we ensure that a 5-year-old gets an online class? But I think this is where probably we'll have to err on the side of caution. We don't want 30, 40, 50 children to be sitting in one class and exposing themselves to each other for the next couple of months. So it's a tough one, but I think it's we are all going through tough times. So I would say yes, hold on for a couple of days, make as much use of technology until we have the right solution.
Okay, okay. So we have an interesting question coming in from Mr. Advait. Um, so we have read about news reports about a COVID person who has already been treated and tested negative for COVID,
Hmm.
again tested, again contracting the disease after a couple of times. So
Hmm.
is it something to do with the diagnostic error, or is it something to do with something else, which is why he's
Okay.
carrying this virus again and again?
So there could be a couple of reasons. See, if someone is tested and that person is negative for the first time, so obviously we're not 100% sure that whether that person is having COVID or not, because the symptoms of COVID and a regular flu are fairly similar. Number one. Number two, okay, it's acceptable that the person did have COVID in the first round. When he or she was tested, that was very early days, or that was after the viral phase, wherein the virus counts had gone down. It could be poor procedure. It could be non-collection of sample. Maybe how the sample was collected and transported was not good. Maybe it was the actual test kit which was wrong. So there could be many factors to get a negative result. Having said that, it is yet to be proven that when a person is infected with corona, that person cannot be reinfected. It's there's no clinical evidence of that. So I know there's a lot of talk about herd immunity, that if 70%, 80% people get infected, then maybe then those people will never have corona again. It's yet to be proven. It's very early days. Epidemiology itself takes years, and we over here we're talking on weeks. So it's quite possible that a person is infected. A couple of weeks later, that person may be infected again.
So, see, doctor, so the PCR test is often comparatively time consuming considering the population that we have, which
Hmm.
has to be tested. So, Kerala government is also planning for a pooled testing.
Hmm. Yeah.
So, could you please give us an overview about the pool testing? Why is it so? Why does it seem to be more less time consuming than, let's say, PCR? And is it that
Mhm.
effective?
So that's a good question. In fact, we also read a few papers about pool testing. So the idea is that, see, PCR by itself is a sensitive test. So whether you test the sample of one patient at a time, or you test the sample of, say, four patients mixed together at a time, on theoretically, I don't know about what happens practically. Theoretically, even that one, for example, you take four samples together. Out of that four, only one person was infected, the rest three were not, right? So you'll still get one positive, one result. Or there's a bigger chance that all four of them are negative. So your pool test will give you a negative result, and in that way, you save resources and you save time, because by doing the test of four together, you've saved the various consumables and the number of test kits. So as a concept, there are some people who are talking about it, especially when you're talking about people with no symptoms at all. So it's possible that in a say in a college, for example, there was one person who was infected, but ideally, yes, then you have to screen all the remaining students because they might be infected, or they might have been exposed to the person who's infected. So instead of testing each and every individual once, the concept is to then do pool testing, because these people are not symptomatic anyways. So it's a concept I've come across. I can't give a lot of expert comments on it because I've not myself personally tested on it, or as a company we have not. But that looks one way, and I'm not against it. Maybe that's a good way of fast turnaround time and saving resources. Maybe yes.
So, since you were involved with the approval process as well,
Hmm.
so we would like to know, let's say, for non-invasive tests
Hmm.
that require an approval from the FDA as well.
Yeah. So all diagnostic tests in India require clearance from FDA, which is the Food and Drugs Administration, and also CDSCO, which is the Central Drugs Control Organization, Standards and Control Organization. So for a lot of these sensitive assays, like I was talking about HIV, TB, malaria, syphilis, and now COVID also, they have certain designated centers. So they would tell you that you have a test, okay, go and get it tested at this particular center. So they will let us know, okay, and those are usually government centers. Okay, for example, uh, in 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 Delhi, there's something known as NIB, which is National Institute of Biologicals. There's something known as the ILBS, which is Institute of Liver and Biliary Sciences. So these people give the report back to the CDSCO. For some other essays which are not that important, the government of India has given approval that you can go to an NABL accredited lab. It could be a private lab also, but that particular lab should have that test in the scope of its accreditation. So if I want to test, say, for the presence of a particular cancer, I can go to a lab which has an NABL accreditation and which has included that particular cancer test in its scope. And then I can get a validation done from that lab and submit that report to the FDA or the CDSCO. So there are very clear cut guidelines given by the Center for these non-invasive tests.
So, does your company make antibody testing kits as well?
No, we are totally focused on molecular diagnostics. Antibody or rapid tests is not our domain.
Okay, so does it have any relevance with the current protocols that are being followed?
That's a question a lot of people have been asking, and I think I think it's a mixture. A group of people whom we are suspecting to be currently infected can be tested on PCR. Someone who said that he had infection, say 15 days back, or someone who says that he was exposed to someone 15 days back, maybe he or she can undergo an antibody test. What an antibody test will tell you, if that comes out to be positive, is that this person has had COVID in the past. Okay, that again does not mean that he cannot have it in the future, or that does not obviously mean that this person is not actively suffering from COVID. So the antibody test is important, but it comes with certain riders. It cannot be taken as a positive negative test.
So one thing which we found quite surprising is that we all believe that the virus emanated from Wuhan.
Hmm,
So the country which is very close to the start of epidemic was Taiwan.
hmm,
But it seems Taiwan never had a lockdown as severe as other countries. Taiwan is back to business. Schools are open.
hmm,
Business is running. Um, yeah. So what makes it? I mean, since it is very close to the epicenter of the epidemic, but still they are back to business. I mean, everything is back to normal. But we have countries which are far away, like France, India. We are still struggling to restart our economy. So, what makes
hmm.
Taiwan a different altogether?
Arun, I'll be very frank with you. I've not read a lot about Taiwan, but from just putting two and two together, what I also understand, Taiwan was one of the first countries to alarm the world about the presence of certain infection in China. So maybe in the earlier days, I'm talking about uh, late December, early January, they probably at that point of time ensured that there was very little travel between that Wuhan province and Taiwan. Again, I have not read much about it, so it's better for me not to make a comment on that.
So, we have one interesting question from Ms. Rakhi. So, she would
Mhm.
like to know future research opportunities for students in molecular biology.
Right. I think that's a very interesting topic. See, as far as molecular biology is concerned, is a very, very new topic to India. It's been around in the West. So there are many diseases, something say like a thalassemia, a sickle cell disease, or for that matter, infection like say a TB, malaria. So there are some infections, some diseases. There are some specific cancer, like oral cancer is very, very prevalent in India as compared to other parts of the world. So I think there's a lot of scope for molecular biologists in clinical diagnostics in India to understand what are the particular mutations which are occurring commonly in India, what particular diagnostic tests are relevant to patients in India. So yes, I would definitely encourage a lot of people to take up molecular biology. That's the future of clinical diagnostics. That's the future of pharmacogenomics, because drugs are going to be based on the mutations that you found. It's not going to be a one size fit all drug. For example, a simple example I'll tell you is TB. So for treating TB, now we look for the resistance of that particular antibiotic in a patient, and that resistance is checked by looking for a particular mutation in a gene. And this is just a very simple way of telling you the same thing could apply to malaria. The same thing could apply to any infection. And with the high population in India, with the number of higher infections in India, there's a lot of scope.
So, coming as it is towards end of our lockdown, the next phase of this phase of lockdown. So, the governments are planning to open up air travel, the rail travel to some extent. So,
Hmm.
would all the gains that we have earned in this lockdown in terms of controlling the infection be
Hmm.
wiped out in a single day once the lockdown is over?
It's some questions are very difficult to answer. It's also important to ensure that families are getting back to where they should be. It's important for businesses to start moving. So it's very difficult for me to answer. I'm not an expert on that particular field. But it's a tough one. We have to balance it out. We have to take very good history. Something like an Arogya Setu app. I personally have not really dwelled into that, but why not? If you get to know that a person has traveled to a certain area where there was a large number of cases of COVID, maybe then that person should not be allowed to board a flight. Again, these are just I'm not an expert, but at the same time, if you see from the history of that person that he's not traveled to an exposed area, then why not? That person should be allowed to move. So it's going to be a mixture. Uh, we've done a great job so far, and let's believe that we'll continue to do a good job in the future also.
Okay, okay. So another interesting question that we have is that the countries like US, where they claim to have a really advanced healthcare facility, seems
Hmm.
to be at loss for any answers at this point of
Right.
time. But then countries like our country, like
Hmm.
India, we have been with limited amount of investment into our primary health care. We have been able to have to face a good fight against COVID.
Hmm.
So where did they actually go wrong? Because in US, if you recollect, they have a pretty robust healthcare management system as well.
Hmm.
So they could have easily integrated their immigration data into the healthcare system, and to some extent, using technology, screen the passengers and people
Hmm.
uh,
Hmm.
in order to ensure that spike in the death rate didn't happen. But then things never happened in that way. So what really happened? Would you be able to answer?
See, again, my personal perception over here is that could be multiple factors. It's not just one answer fits all. So obviously, China was the first one to be exposed. European countries didn't really understand how big could this be. Places like Italy and France, in early February when they were exposed, they just thought it's one more virus. It'll spread like H1N1. Few people might die. No one really understood how virulent would this be. India was lucky in that sense because by the time it came to India, middle or late of March, we knew the natural history of the disease. We knew how it spread, right? So that's one factor wherein we had the advantage. I also read about the age profile. I also read that Italy probably has one of the highest median age of population. So that was one factor over there. Then there might be other things as well. So one place where another article which I read in the BBC, which read that a lot of these countries are not prepared for an epidemic now. They've kind of developed the healthcare system in a way that those things don't happen to them. So they were not ready to face 100 people entering an emergency department with breathlessness, which is unfortunately not the case in India. I'm a doctor myself. I worked in medical colleges. We are used to handling hundreds of patients. It's a regular process for us. So a lot of these factors might have helped. So that's what I was trying to say, that multiple factors are involved. Yes, Indian government did a great job. We were very proactive in ensuring that there was a lockdown put in place, but we also had the benefit of hindsight, which probably the Western countries did not have. Within 15 20 days of the virus coming out of China, and China obviously did not make a lot of information public, so they didn't know about this particular virus. But by the time it came to India, because Europe and America was highly affected by it by then, we knew a lot about the virus as compared to what Europe and America knew in early February.
Okay, okay. So we have another interesting question from Ms. Nidhi. So here goes the question. So as a doctor, uh, how easy it was for you to translate your knowledge and experience in the field of clinical medicine into molecular diagnostics?
Right. So as I was telling you, it's a team. Our team in my lab is not only I'm not the only person who's involved in R&D. There's a huge team which looks at clinical the R&D side of it. So as a doctor, when I work in an organization like this, or I'm a part of an organization like this, with the perspective that I can bring forward is the magnitude of the problem. With my colleagues, with my interactions with a lot of doctors, we understand how big this problem is. It's important for us to then pass it on to the R&D team to start developing essays around this. COVID is one example, but there are many other things that we talk about. For example, My experience in transfusion medicine tells me that there are many cases of bacterial contamination of blood. Right now, this is something I bring to the R&D lab of, say, my lab. Then we sit down and say, okay, if there's that's one problem area, what can we do to detect the presence of bacteria in blood? So, getting the clinical, experience from interactions and talking to colleagues and visiting various conferences is brought to the R&D lab, and then we sit in the R&D lab and we come out with a solution for that. So it's a mixture. It's a team. We work as a team. So that's what I bring as a doctor.
Okay. So we've been recently hearing a lot of reports about the COVID testing. Since the test kits were not available initially,
Hmm.
it seems that technologies like CT scan
Hmm.
can be used for initial detection of COVID, but it does
Hmm.
not. I mean, it will not give a 100% accurate result. But then,
Hmm.
since the kits were less in supply,
Hmm.
perhaps CT scan can be used as the first level of screening, and
Hmm.
then administer PCR-based tests. So, is it really a scientific approach, Doctor?
See, a CT scan is usually done to see the condition of internal organs, to see how badly they're affected, how badly they're inflamed, or if they're bleeding or something like that. So the the problem with COVID is that there's an infection. Not all of us will react to that infection in a particular manner, but some of us who do will have a very clear picture in the lungs, and that's where a CT scan or a good imaging technique will be able to help. So what a technique like that would be able to do is to select or identify problematic patients who might require a ventilator or tertiary care or ICU support. But to identify whether the person is infected by COVID, I think you still will have to rely on in vitro diagnostic assays like the PCR or something else that might come up. What a CT scan or other imaging assays will do is to identify internally if a person, whether he or she is infected or not, is developing a respiratory or a lung or any other organ related damage because of whatever infection. So it's a combination, right? But a CT scan usually cannot be used as a first line test for any infection. It's going to tell you the outcome of the infection in the body.
So I think we have had a wonderful session. I'm sure that
Yeah.
we have had a wonderful session. It's been quite insightful session. I mean, it's been a great blessing, Doctor, having you here for the webinar. So let me take this opportunity on behalf of Kerala Startup Mission, the Thai Kerala, GTech, which is a group of technology companies in Kerala, and Kerala Technology University for giving us this amazing session, taking time off for the session. Thank you so much, sir, for being kind and graceful for the session. It's been a pleasure having you here, sir. And we would
Thank
also
you.
like to invite you to Trivandrum. We'd like to invite
Mhm.
you to Kerala. Do come here once the
Sure.
COVID situation improves. Our entrepreneurs, our institutions, educational institutions, our research organizations are all looking forward to work with you collaboratively so as we can together do things for the betterment of mankind.
Thank you, Arun, and thank you, Arya, and all the people who've been sitting with us for some time. And yes, why not? We can definitely take it forward soon and see you in person soon sometime.
Thank you so much, sir.
Okay? Thank you. Okay, bye.